Prothero, bringing fossils to life. Introduction to Paleobiology. The fossil record. In the mountains of Parma and Piacenza, multitudes of rotten shells and corals are to be seen still attached to the rocks, and if you were to say that such shells were created and continue to be created in similar places by the nature of the site and of the heavens, which had some influence here, such an opinion is impossible for the brain capable of thinking, because the years of their growth can be counted on the shells, and both smaller and larger shells may be seen, which could not have grown without food and could not have fed without motion, but they could not move. In the mountains of Parma and Piacenza, multitudes of rotten shells and corals are to be seen. In the mountains of Parma and Piacenza, multitudes of rotten shells and corals are to be seen, still attached to the rocks. And if you were to say that such shells were created and continue to be created in similar places by the nature of the sun and of the heavens, which had some influence there, such an opinion is impossible for the brain capable of thinking, because the years of their growth can be counted on the shells, and both smaller and larger shells may be seen. Okay, and if you were to say such shells were created and continue to be created in similar places by the nature of the site and the heavens, and had some which had some influence there, such an opinion is impossible for the brain capable of thinking. Such an opinion is impossible for the brain capable of thinking, because the years of their growth can be counted on the shells, and both smaller and larger shells have been seen, which could not have grown without food and could not have fed without motion, but then there they could not move. And if you wish to say that it was the deluge which carried these shells hundreds of miles from the sea, that cannot have happened, since the deluge was caused by rain, and the rain naturally urges rivers on towards the sea, together with everything carried by them, and does not bear dead objects from seashores towards the mountains. And if you would say that the waters of the deluge afterwards rose above the mountains, the movement of the sea against the course of the rivers must have been so slow that it could not have floated up anything heavier than itself. What is a fossil? When we pick up fossils in a road cut or see a dinosaur skeleton in a museum, we have no problem connecting it to some sort of extinct organism. We have been conditioned since our early education to interpret fossils as remains of extinct organisms, and it is hard for us to imagine any other explanation. Centuries ago, however, such an interpretation was not automatic or even easy to make. The ancient Greeks interpreted the giant bones of mammoths as the remains of mythical giants, but were puzzled with seashells found hundreds of feet above sea level and miles inland. Had the sea once covered the land, or had these objects grown within the rocks like crystals do? In the 6th century BC, Xenophons of Colophon saw the Seychelles high in a cave on the island of Malta and suggested that the land had once been covered by the sea. The oldest recorded statement that fossils are the remains of once living animals that have been included in rocks was made by Xanthos of Sardis around 500 BC. Aristotle, born 384 BC, suggested that the fossils of fish were remains of sea animals that had swum into cracks in rocks and were stranded there, and his ideas were influential for the next 2000 years. From the late days of the Roman Empire, almost all people in Western society were raised to believe in the literal interpretation of the book of Genesis, and the stories of the 6 days of creation and Noah's flood colored their view of rocks and fossils. For those of us in the nearly 21st century, a fossil-made shell looks so similar to its living descendants that we cannot imagine any other explanation. We forget that most people of that time, other than fishermen, had limited familiarity with life on the bottom of the ocean. In fact, many fossils bear no resemblance to anything that 15th century Europeans had could have seen until the living chamber nautilus was discovered in 1829. Until the living chamber nautilus was discovered in 1829, who could imagine that the coiled objects known as Cornua ammonis, horns of ammon, serpent stones, were relatives of the squids and octopus with a coiled and shapered shape? Who could imagine that the strange bullet-shaped objects known as belemnites were also related to squid? Even today, most people would who pick up the odd symmetrical objects known as clinoid columns. Figure 1.2 do not recognize them as relatives of the sea star or sea urchin because only a few people have seen the rare stalked crinoids that still live on the sea floor. For centuries, scholars were impressed with the star-shaped patterns in the centers of the columns and the radial patterns in fossil corals and thought they had been produced by thunderbolts or fallen from the sky. They were known as star stones, lapis stellaris or asteroides stellaris. During the Middle Ages and Renaissance, learned men began to speculate on the meaning of fossils, producing a wide range of interpretations. Originally, the word fossil, from the Latin fossilis, dug up, applied to any strange object found within a rock. These included not only the organic remains that we call fossils, but also crystals and concretions. Okay, these included these included not only the organic remains that we call fossils, but also crystals and concretions and many other structures that were not organic in origin. Most scholars thought that fossils had formed spontaneously within the rock. Those that resembled living organisms were thought to have cracked or fallen into cracks and then converted into stone. Others thought they were grown in rocks from seeds or were grown from fish spawn washed on both his own collection and those of his friend, washed into cracks during Noah's flood. Okay, so it is the Conrad Gessner's 1565 illustrations of bullet-shaped belemnites and crinoid columnals, neither of which looked like marine organisms familiar to Renaissance Europeans. To many of them, including Gessner, the star-like pattern of some crinoid stems suggested that they might have been produced by fallen stars. Many scholars thought they were supernatural pranks of nature, lustas naturally, of figured stones produced by mysterious plastic forces. Still others considered them to be works of the devil placed in the rocks to shape our fate. As quaint and comical as these ideas seem to us today, in their own time, they were perfectly rational for people who believed in literal interpretation of Genesis and thought the earth had been created just as we see it about 6,000 years ago, with little or no change except for decay and degradation due to Adam's sin. Some Renaissance men, however, were ahead of their time. Around 1500, Leonardo da Vinci, 1452 to 1519, recognized that the fossil shells in the Apennine Mountains of northern Italy represented ancient marine life, even though they were miles from the seashore, unlike his contemporaries who thought they had been washed there by the flood. Da Vinci realized that they could not have washed that far in 40 days, and many shells were too fragile to have traveled that far. Many shells were intact and in living position and resembled modern communities found near the seashore. Clearly, they were not transported. In some places, there were many shell beds separated by unfossiliferous strata, so they clearly were not due to single flood. However, most of Da Vinci's ideas remain in his unpublished notebooks, and even if he had tried to publicize them, they would not have been accepted at that early date. In 1565, the Swiss physician Conrad Gessner, 1516 to 1565, published De Rerum Fossilium, or the nature of fossils, the first work that actually illustrated fossils. With this state, the vague verbal descriptions of earlier authors could be made more precise. Gessner based his descriptions on both his own collections and those of his friends, beginning the modern tradition of scientific exchange, analysis, and comparison. Gessner was correct in comparing most fossils to their living relatives, but he thought that some objects, crinoid, belemnites, were formed by mineral precipitation. Like most of his contemporaries, Gessner interpreted fossils as the supernatural representations of Neoplatonic ideal forms and did not explore most of the implications that would seem obvious to us today. Through all of these early writings, four main questions about fossils were disputed. Are fossils really organic remains? How did they get into the rocks? And when did they get there? As the rock was beginning being formed or long after? How did they become petrified? Essentially, modern answers to all these questions were proposed by a Dane named Niels Stenson, known to the latter generations by the Latinized version of the name Nicholas Steno. Steno was the court physician to the Grand Duke of Tuscany, so he had ample opportunity to see the shells in the rocks of the Apennine Mountains above Florence. In 1666, he had a chance to dissect a large shark caught near the port town of Livorno. A close look at the mouth of the shark showed that its teeth closely resembled fossils known as tongue stones. Glossopetri Petri, okay, which had been considered the petrified tongues of snakes or dragons. Steno realized that tongue stones were actually ancient shark teeth. Steno realized that tongue stones were actually essentially actually actually as ancient shark teeth, and that fossil shells were produced by once living organisms. In 1669, Steno published De Solido Intra Solidum Natural Liter Contento Desert Shanis Podrumas. Okay, forerunner to a dissertation on a solid naturally contained within a solid. The title may seem peculiar at first until you realize the central problem that Steno faced. How did these solid objects get inside the rock? Steno realized that the enclosing sandstone must have once been loose sand and was later petrified into sandstone. With this idea, he overturned the long-standing assumption that all rocks have been formed exactly as we see them during the first days of creation. Steno extended with this idea, he overturned this long-standing assumption. Now, Steno extended this insight into a general understanding of relative age of geological features. Fossils that were enclosed in rock that had been molded around them must be older than the rock in which they lie. On the other hand, crystals that clearly cut across the pre-existing fabric of a rock must have grown within the rock after it formed. From this, Steno generalized the principles of superposition, original horizontality, and original continuity that are the fundamental principles of historical geology and stratigraphy. As the Prodromus was being published, Steno converted to Catholicism and gave up his scientific interest, so the forerunner was never followed up by the promised dissertation. He was eventually made the bishop of Titiopolis, a region in Eastern Europe that had not converted to Catholicism, so he never lived there or ministered to its people. Okay, so instead, he returned to Denmark to serve the church for the rest of his life. About the time that Steno's writings appeared, A pioneering British scientist came to similar conclusions. Robert Hooke is better known as the father of microscopy because he built one of the first microscopes and made the first drawings of microorganisms and details of cellular structure. In 1665, Hooke made observations of many natural objects, including the first accurate drawings of fossils, which were published posthumously in 1705. Hooke even suggested that fossils might be useful for making chronological comparisons of rocks of similar age, such as much as Roman coins were used to date ancient historical events in Europe. He speculated that species had a fixed lifespan, for many fossils he studied had no living counterparts. This was one of the first hints of the extinction of species because few people at that time doubted that all the species on Earth had been created 6,000 years ago and were still alive. However, most of Hooke's and Steno's ideas would not be accepted for another century. In the early 1700s, ideas about fossils were still heavily influenced by the Bible. For example, in 1726, the Swiss naturalist Johann Schuster described a large fossil as the bony skeleton of one of those infamous men who since brought upon the world the dire misfortune of the Dead Age. He named it Homo diluvi testis, man, a witness of the flood. Unfortunately, since this is the Homo diluvi testis. Actually, the fossil of a giant salamander. Okay, unfortunately, since comparative anatomy was not very sophisticated at this time. His specimen turned out to be a giant fossil salamander. Another scholar, Dr. Johann Beringer, 1667 to 1740, dean of the medical school of Würzburg, Germany, was fascinated with the petrifications that collectors had brought him from the local hills. Some bore resemblance to frogs, shells, and many other natural objects. Others had stars and many other curious shapes and patterns. In 1726, as Beringer was about to publish a massive monograph of his figure stones, two colleagues whom Beringer had offended confessed to the prank. They had carved the figured stones, correctly guessing that he was gullible enough to accept them, but their warning came too late to stop publication of the hoax. Beringer died a ruined man, spending his last uh, penny trying to buy back all the copies of his book. By the mid 1700s, however, naturalistic concepts of fossils began to prevail when Linnaeus published the first edition of his landmark classification of all life, Systema Naturae, in 1735. Fossils were treated and named as if they were living animals. Around 1800, Baron Georges Cuvier, 1769 to 1832, made great strides in comparative anatomy, skillfully showing that certain anatomical features such as claws and sharp teeth or hooves and grinding teeth were correlated. He became so adept at this knowledge that he started the paleontological tradition of predicting unknown parts of the animal by comparison to the unknown anatomy of close relatives. Cuvier also showed that the bones of mastodons and mammoths were the remains of elephant-like beasts that clearly had to be extinct because the explorers had not found them on even the most remote continents. Prior to that time, most people could not accept the fact of extinction because it went against the notion of divine providence. After all, if God watched after the little sparrow, surely he would not allow any of his creations to go extinct. Cuvier went on to become the founder of comparative anatomy and vertebrate paleontology and brought much of paleontology out of biblical supernaturalism and into a firm comparative basis. Okay, there is a lot. Just before 1800, British engineer William Smith, 1769 to 1839, was surveying England for the Great um, canal excavations prompted by the industrial revolutions. From these fresh canal exposures and regular visits to mines, Smith began to realize that fossils showed a regular pattern. Each formation had a different assemblage of fossils. As he wrote in 1796, he was struck by the wonderful order and regularity with which nature has disposed of these singular productions fossils and assigned each to its own class and peculiar stratum. Smith was so good at recognizing the fossils of each formation that he amazed private collectors by correctly identifying the layers from which their specimens had come. He used this understanding of faunal succession to map the strata of England and Wales, culminating in the first modern geological map, which was finally published in 1815. At about the same time, Cuvier and his colleague Alexandre Brongniart were mapping fossils and strata in the Paris basin and also began to realize that there was a regular succession of fossils that differed from formation to formation. In two different regions, apparently independently, each made the discovery that eventually led to our modern concepts of biostratigraphy as a tool for unraveling Earth history. By the time of the publication of Darwin's On the Origin of Species in 1859, the realization of the complexity of the fossil record had reached the point where few scholars took Noah's blood literally. However, the notions about what the fossil record tells us about the history and evolution of life has continued to change, as we shall see in later chapters. How does an organism become a fossil? Being a paleontologist is like being a coroner, except that all the witnesses are dead and all the evidence has been left out in the rain for 65 million years. There are over 1.5 million named and described species of plants and animals on Earth at this moment, and probably many more that have never been named or described. Some estimates place the total number at about 4.5 billion million species. Yet, the fossil record preserves only a small fraction of this total and does so in a very selective manner. Some groups of organisms had with hard parts, such as shells, skeletons would tend to fossilize readily, and much is known about their past. Many other others are soft-bodied and rarely, if ever, fossilized. So paleontology has little to say about their history. The study of how living organisms become fossilized is known as taphonomy, Greek laws for laws of burial. There are several ways to get a sense of just how unlikely fossilization can be. For example, modern biological studies show that the typical sea bottom is often dense with shells. One quarter of a square meter of seafloor of Japan, Thorson 1957, yielded 25 individuals of a large bivalve, Macoma incongrua, 160 of a small cockle shell, Cardium hungerfordi, and 12 of the tusk shell, Dentalium octangulatum. The average age of these mollusks is 2 years. At this rate, there would be 1,000 shells in just 10 years, or 100 million in million years. Over one quarter of a square meter, extrapolated over the whole seafloor over geological time, this suggests that a staggering number of shells could have been fossilized. In fact, that tiny area of seafloor near Japan could produce more fossilizable shells than is actually known from the entire fossil record. Clearly, most organisms do not become fossils. The study of taxonomy has become very popular in the last 20 years for one simple reason: to understand and interpret the preserved fossil record. You must first determine how taxonomic processes have biased your sample. From the moment an organism dies, there is tremendous loss of information as it decays and is trampled and broken before it is buried. The more of that lost information that we can reconstruct, the more reliable our scientific hypotheses are likely to be. In this sense, every paleontologist has to act as a coroner or forensic pathologist or detective, determining what killed the victim and trying to reconstruct the events at the scene of the crime. The first step is to determine just what type of fossilization has taken place. Most fossils have been dramatically altered from the original composition of the specimen, and often their original shape and texture are hard to determine unless one had some idea of what took place. The major types of preservation processes are discussed next. Unaltered remains. In a few exceptional cases, organisms are preserved as most of their original tissues intact. Okay, in a few exceptional cases, organisms are preserved as most of their original tissues intact. Ice Age woolly mammoths have been found thawing out of the Siberian tundra. Okay. So, Ice Age woolly mammoth have been found thawing out of the Siberian tundra with all their soft tissues essentially freeze-dried, including their last meals and their digestive tracts. Some were so fresh that humans and animals could eat their 30,000-year-old meat with no ill effects. Some were so fresh that humans and animals would eat their 30,000-year-old meat with no ill effects. My goodness. And Ice Age, an Ice Age woolly rhinoceros was found intact in a Polish oil seep. The petroleum pickled specimen and prevented the petroleum pickled the specimen and prevented decay. These examples are extremely rare, but when they occur, they give us detailed insight into the color, diet, muscles, hair texture, and other anatomical features that paleontologists seldom see. Some fossils have decayed so that the soft tissues are gone, but their hard parts are unaltered. The famous tar pits of Rancho La Brea in Los Angeles are full of 40,000-year-old bones that retain their original composition but are pickled in tar so they are black and smell like petroleum. However, enough of their original chemistry remains that scientists have been able to extract their DNA and compare it to that of living relatives. Similarly, many shells, particularly those of Pleistocene age, still have their original shell material unaltered, including the iridescent mother of pearl aragonitic layer that frequently lines the interior. In a few places, Cretaceous ammonites have been found with their original aragonite intact, but there are few fossils much older than this that retain their original aragonite. Preservation in amber is a special case. These specimens are fossilized when tree resin oozes downward and entraps insects, spiders, and even frogs and lizards. The resin then hardens and forms a tight seal around the organism. Most specimens are only carbonized bones of insects and spiders, but some are so well preserved that their original biochemicals are still intact. Some of the molecules have been successfully extracted and sequenced, as depicted in the movie Jurassic Park, but the genetic material is so incomplete that we will never be able to reconstruct a complete organism. For mineralization, many biological tissues are full of pores and canals. The bones of animals are highly porous, especially in the marrow cavity, and most wood is full of canals and pores. After the soft parts decay, these hard parts are buried and then permeated with groundwater that flows through them. In the groundwater, a dissolved calcium carbonate or silica, which precipitate out and fill up the pores, completely cementing the bone or wood into a solid rock. Unlike replacement discussed below, new material comes in, but none of the original material is removed. The famous multicolored fossil logs of the Triassic petrified forest of Arizona are permineralized by silica, and many other examples of petrified wood and bone are permineralized by carbonate. Permineralization can be so complete that even the details of the cell structure are preserved. Recrystallization. Some shells are made of relatively unstable Minerals such as aragonite, once the shells leave surface conditions, most aragonite reverts to the more stable form of calcium carbonate, the mineral calcite. In other cases, shells made out of tiny crystals of calcite recrystallize into larger crystals. In these cases, the original shape of the fossil is preserved, but under the microscope, the difference in the texture is apparent. However, the chemical composition is unchanged. Dissolution and replacement. As water seeps through sediments filled with shells or bone, there is also a tendency for the original material to dissolve. If the fossil dissolves and leaves a void, then the shape of the fossil is preserved in the surrounding sediments. The internal filling of this specimen is known as an internal mold or stone cast in German. Okay. The external mold of the specimen is often also preserved. In other cases, the void is filled with sediment and a natural cast of the fossil is formed, mimicking the original in surprising detail. Original bone or shell material can also be replaced without leaving a void. In these cases, the original calcite, aragonite, or phosphate is dissolved away and another mineral precipitates almost immediately in its place. This is easiest to detect when a fossil is made of some mineral that is clearly not original, such as calcitic brachiopods now made of silica or pyrite. Okay, so see the internal mold. This is the internal mold, stain card. Okay, the cast. This is the external mold, recrystallized carbonate or replaced mineral, original skeleton material. This is the original skeleton material, original void. This forms a carbonate matrix, and this forms the void fitting cement. This forms sandstone filling, then fossil dissolves away, and then new mineral precipitates into the void by the fossil. Then this carbonate dissolves away, leaving crystallized void filling. Okay, so this is taphonomy. Diagenetic processes, not taphonomy, diagenetic processes. The external mold of the specimen is often also preserved. In other cases, the void is filled with sediment and a natural cast of the fossil is formed. Mimicking the original in surprising detail, original bone or shell materials can also be replaced without leaving a void. In these cases, the original calcite, aragonite, or phosphate is dissolved away and another mineral precipitates almost immediately in its place. This is easiest to detect when a fossil is made of some mineral that is clearly not original, such as calcitic brachiopods now made of silica or pyrite. Carbonization. Many fossils are preserved as thin films of carbon on the bedding planes of sandstones and shells. When the organism dies, most of the volatile organic materials disperse and leave a residue of coal-like carbon in the form of a black film that preserves the outline and sometimes the detailed structures of the organism. This kind of preservation is typical of most plant fossils. Indeed, coal is the accumulated carbonized films of countless plants. This kind of preservation is typical of most plant fossils. Okay, indeed, coal is the accumulated carbonized films of countless plants. There are many examples of carbonized animal fossils, especially among the graptolites. They are that are virtually always preserved as carbonized films. There are also Eocene fossil insects preserved in extraordinary quality in places such as Florissant, Colorado, and the body outlines of ichthyosaurs from the Jurassic Holzmaden shale in Germany. We will keep it till this much and continue in the next part.